Bismillahirrahmanirrahim. Assalamu alaikum. I'm here from Conceptual Medicos, and today in this lecture we will be talking about the genus Vibrio. It has three main important species, which are Vibrio cholera, Vibrio parahemolyticus, and Vibrio vulnificus. In these, Vibrio cholera is most important exam point of view, so we will mainly focus on Vibrio cholera. The Vibrio parahemolyticus causes diarrhea, and Vibrio vulnificus causes cellulitis. Okay. Now the combined features of this genus is they are curved organisms, they are comma shaped, and they are gram negative organisms. Okay. The other features are they are oxidase positive, unlike the enterobacteriaceae which are oxidase negative. Okay. So coming towards our main topic, which is Vibrio cholera. So Vibrio cholera, on the basis of O antigen, they are subdivided into serogroups. So what is O antigen? This is basically a cell wall antigen. The other antigens and different gram negative organisms, gram negative rods, are basically the F antigen, which is flagellar antigen, and the K antigen, which is the capsular antigen. F flagellar and K capsular. Okay. So on the basis of O antigen, they are divided into serogroups, different serogroups, among which O1 and O139 are very important, and these are important because they produce toxins. And due to O139, there was epidemic outbreak in 1992. The O1 is very important, and it is divided into subtypes which are known as biotypes of O1, and the one is Iltor and the other one is Glossic. Okay, coming towards the transmission, so basically the Vibrio cholera is transmitted by fecal contamination. Fecal contamination of water and food, and basically the animal reservoir are marine shellfish like shrimp and oyster. So when the water or food is contaminated by the human feces, and when we eat or drink such type of food and water, then it enters into our body and causes the disease there. Okay. Coming to the epidemiology, so it has very important regarding this topic that the cholera epidemic occurred in 1970 in Asia, and it spread into the Africa and then into the Europe. In 1991, the outbreak occurred in America. In 1992, the outbreak occurred in India, and the notorious one was Iltor, which is a type of serogroup O1 of Vibrio cholera. Now the predisposing factors are poor sanitation. So when there is poor sanitation, there will be no proper disposal of the feces of humans, so the water will be contaminated and it will be easily affecting the human beings. Okay. The next one is overcrowding and inadequate medical services. Coming toward the pathogenesis, which is very important in exam point of view. So a very important point is it is sensitive to stomach acid, and means large amount of organisms are required to cause cholera in a human being because they are sensitive to stomach acid. So the patient which are on anti-acids or which have done gastrectomy, so they are easily affected by this organism. Okay. Now adherence, the second step is adherence. So adherence occurs through villi to the brush border of the small intestine. Mucinase is an enzyme which have a role there. It basically dissolves the glycoprotein over the enterocyte and allow the organism to adhere there, and colonization occurs then. Next one is the production of the enterotoxin. The enterotoxin is known as the cholera toxin. Coming towards the mechanism of the cholera toxin, that how it works. So basically it consists of two parts, the A part and the B part. The B part is basically the binding subunit, and the A is basically the active subunit which will do the action. Okay. It comes there, the B part help in the binding, the A part is inserted inside. The A part causes ADP ribose to bind with the G stimulatory protein. G stimulatory protein is a second messenger basically. So by activating this GS, it will causes the activation of the adenylate cyclase enzyme, and it will cause the conversion of the ATP into cyclic AMP. So when the level of the cyclic AMP rises, it activates basically protein kinases, and when protein kinases are activated, it do the phosphorylation of the chloride channels. So the chloride channels open, and there is loss of chloride channels followed by water, which result in massive watery diarrhea. Okay. Coming towards the clinical findings, so basically the clinical finding is a watery diarrhea, which is very severe, and a very special term is used here, which is important for the exam, that rice watery stool are observed in this condition. Okay. No neutrophils or RBC are there in the stools. And why is it so? Because this is basically a toxigenic organism, not an invasive organism. And the invasive organism, we observe neutrophils in the stools. Okay. There will be no abdominal pain, as in many diarrheal cases and caused by other organisms, we have abdominal pain. There will be dehydration because of loss of very much amount of water. Okay. Now the complication will be cardiac failure and renal failure. This is because of hypovolemia due to loss of water, and acidosis because of loss of bicarbonate, and hypokalemia because of the loss of potassium ions. Okay. Now coming towards the lab diagnosis, so in epidemic cases, we just do the diagnosis by clinical judgment. If a person has watery diarrhea and dehydration and such type of symptoms, then we do the treatment on these clinical judgment. And endemic cases, we do the TCBS agar culture. Okay. And sporadic cases, sporadic means if the recurrent infection occur in a person, we do stool culture on McConkey agars. So this organism is a slow lactose fermenter, so colorless colonies are observed. Okay. This organism is oxidase positive, unlike the Enterobacteriaceae, which are oxidase negative. On TSI agar, the slant is acidic and the butt is also acidic. This is because of sucrose fermentation. Okay. The next one is the agglutination. The patient sample is mixed with O1 anti-sera. If the agglutination occurs, means the toxins are present. The treatment is basically the adequate replacement of water and electrolyte. No antibiotics are needed basically. Okay. So the antibiotics, if we give antibiotics to the patient, they will just increase the rate of excretion of the organism from the gut. Prevention by clean water plus food and Pexcora vaccines are available but not usually used.